everyone. Thank you so much for joining the NepSciHub this evening for yet another episode of COVID-19 Conversations. We received great feedbacks from our previous conversations with Dr. Daniel Griffin and Dr. Monica Gandhi. And in our quest to bringing you engaging and stimulating contents pertaining to the pandemic, we've amongst us another distinguished guest, Dr. Alba Grifoni. Dr. Grifoni is an instructor and research faculty at the Center for Infectious Diseases and Vaccine Research at La Jolla Institute for Immunology in La Jolla, California. She'll be speaking about the important role T cells play in viral infections and the lessons that we've learned so far from COVID-19. Her publications have been fundamental to understanding of SARS-CoV-2 immunology. Her paper, Targets of T-cell Responses to SARS-CoV-2 Coronavirus in Humans with COVID-19 Disease and Unexposed Individuals, was one of the highly cited papers, and it was widely covered by mainstream media like the New York Times and the Washington Post. Such was the influence of that publication that even Dr. Anthony Fauci presented it at a Congress meeting. Her works have also led to a deeper understanding of variants and their impacts on T-cell recognition. In recognition of her tremendous works, she was recently received the Embassy of Italy Award to honor young Italian researchers to research to fight against COVID-19 from the Italian Scientists and Scholars in North America Foundation, ISS, NAF. As usual, the format of tonight's webinar will be a presentation by Dr. Grifoni, followed by a Q&A from your audience. So I'd just like to quickly remind our audience to use our chat box. We'll try to address as many questions as possible. Without further ado, I'd like to thank Dr. Grifoni for your time. We really appreciate it. I hope that tonight's webinar will be absolutely informative, just like the last two. Dr. Mahat will be our moderator and facilitator. Dr. Sargama, the floor is all yours. Thank you so much.
So thank you very much. This is a nice invitation, and I'm really happy to be here and to start this chat about T cells. I guess from the presentation that you received, you might understand that I am T cells oriented. I'm in love with T cells, always been. I've been in the La Jolla Institute for Immunology with Professor Seth and Weiskopf laboratory since 5 years. And throughout my careers, I've been interested in understanding how T-cell response recognize viral infection. And then in 2020, SARS-CoV-2 hit us, so we decided to actually understand better what was going on with this virus. But just to give you a background about T cells, because for many people that maybe are not expert in the area, T cells are kind of a mystery immunological population. So if we put a little bit in the context of what we mean with an immune response in general, the immune response is composed by two different major components. One is defined as the innate response, and that is usually the first line of response. As soon as you get an infection, the innate response doesn't really recognize specifically what is going on, but they basically use some kind of like alarm bells based on type 1 interferon gamma expression. And what they try to do is to basically limit the viral replication in order to reduce the spreading of the virus. Another important part of the innate immune response is to recruit effector cells and mostly prime the adaptive immune response. And the adaptive immune response is actually the component of the immune system that is considered a second line of response. And this response is actually specific for the virus. So it takes time to actually be able to build appropriately. But the advantage of the adaptive immune response is that once they encounter the virus, they remember it. So they develop what we call memory of the immune system. And the developing of this memory is critical to actually design a vaccination, because this means that the adaptive immunity, once they encounter the virus or something that resembles the virus, such as a vaccination, they remember it and they are able to respond faster and specifically to limit the infection. So the adaptive immunity overall is composed by different components, and the main actor of this immune response are actually three: the antibodies response and the T cell response. So So while the antibody maybe don't require as much presentation because they're fairly known elsewhere, I'm gonna dig a little bit more on the T cells. But to do so, I would like to give you like a difference between what antibodies recognize and what T cells recognize. So the antibodies is a response that is actually, those are proteins produced by B cells, and they're able to recognize proteins that are located only on the surface of the virus. But once the virus infects the cells, the antibodies don't see them anymore because they only recognize what is on the surface. So this is where T cells kicks in. So there are two different main types of T cells. The first one, they are CD8 T cells, also known as killer T cells. And what do? Is that they do patrol the entire body and they basically scan the different cells to identify cells that are infected. And if they are, the killer T cells kill them. That's where they take their own name. The CD8 T cells killers are able to recognize if a cell is infected, and they recognize any type of cells that has a nucleus. While a different type of T cells, which are the helper T cells, those are specialized type of T cells. They are able to not only recognize that an infection is going on, but they also recognize the type of pathogen, and they understand what is the best quality of immune response that is required to efficiently control the infection. This is why they act as a coordinator of the entire immune system, and they orchestrate both the response of the killer T cells as well as the antibodies response. And in fact, they help and they are critical for the antibody response in particular. So this is one of the main difference between the two. As I was mentioning, antibody recognize only the external part of the virus, while the T cells virtually are able to recognize the entire virus. So with this concept in our minds, let's try to apply this knowledge to SARS-CoV-2. And I believe many of you already know a lot about this virus. So I'm just gonna quickly remind you what is the protein composition of the virus. So the SARS-CoV-2 is a coronavirus, part of the beta coronavirus, and is composed by structural proteins that are located on the external side of the virus. The major one, the spike glycoprotein, that is composed by a trimer. This protein is very important for the virus to enter inside the cells. This is why it's the main target of the antibodies, and antibodies that are able to bind to the spike and prevent infection, they do what is called neutralization. There are other type of proteins that are located in the surface of the virus, such as the nucleocapsid protein. Then in fact, we do know that there are antibodies against this protein as well, and the membrane protein, as well as an envelope protein. So all these structural protein protects what is the genome of the virus, which once is inside the cells, it starts replicating itself and produce several different type of proteins. What we call the genomic RNA, encoding what we call them non-structural proteins. So those are proteins that help the virus to replicate, but are not correlated with the structure of the virus. And then there are the different type of proteins, which are the ones that I already described to you, that are focused on composing the structure of the virus that allows the virus to exit the cells and infect another one. So antibodies, as I was mentioning, they recognize only what is on the external side of the virus. And in fact, they recognize mostly the spike protein. And this was the rationale behind the decision of designing at the real beginning of this epidemic, vaccination against the spike protein. Because we know the spike was the surface protein. We know that it was recognized by the antibody. And at the time, we didn't know if this protein was also recognized by the T cell response. Conversely, the T cells have virtually the capability to recognize all the protein of the virus. In fact, once the virus infects the cells, the proteins are processed and presented in the context of HLA molecules. And I'm gonna go over the specific of the molecular recognition of T cells and HLA later on this talk. But just right now, to follow the next slide that I'm gonna show you, just understand that the T cells do not recognize the entire protein, they just recognize pieces of it that are presented in the context of these molecules. So the question that we wanted to have, and the way I'm gonna try to guide you through is actually the science that we had during the beginning of pandemic. So imagine an outbreak comes, you know nothing about this virus. You don't know even if there is a response against this virus because it's novel, nobody has ever studied. So the first very naive question, perhaps at the time, but very critical is, do we have an immune response that is actually gonna recognize this virus? Meaning, can a vaccination approach is gonna work in this case? So we decided to do that, and the cohort that we decided to study in this case was a cohort of subjects that experienced the infection, they got COVID-19, very mild, they recovered from it. So they kind of represent the majority of the population that usually get infected with the virus and has an asymptomatic or a very mild symptom disease. And when we did that, what we saw is that 70% of our COVID-19 donors were having actually a very strong CD8 killer response. So this was great, but that was not sufficient. We were, what else? What about the CD4 T cell response? Because that is very important for the antibody. And we were happy to see that also in the context of the CD4 T cells helper, we were having a significant response in spike protein. Actually, all the donors that we tested were responding against the spike. And the response was not only directed against spike, but also against all the other proteins of the virus. So overall, what does this mean? This means that there were antibody response. And of course, in our study published in Cells, we also evaluate the antibodies. But the good news here was we have the three components of the immune system all reacts against spikes. So this is great. It means that the vaccination strategy is successful, it's going to work. And indeed it worked, as we know right now. So one of the questions that we were asking was, okay, so there is a strong response against spike, but this is not the whole story. So what are the proteins are T cells able to recognize? So we decided to actually look at each one of the different proteins that are composing the virus. And here I'm going to show you just the example for the CD4 of this paper. And you can appreciate that the response is not always the same in all the protein, but it's actually protein dependent. With the major protein recognized are the structural ones, membrane, nucleocapsid, and spike, followed by others, also non-structural protein. So the first evidence that we saw in trying to understand how does this response work was that the strength of the response was actually related to the abundance of the protein. Because clearly, when the virus replicates, it doesn't need that many non-structural proteins. I mean, hey, I need a couple just to replicate myself, but what they really need is the structural proteins to actually perform exit from the cell and infect a new one. So naturally, the structural proteins are more abundant, and therefore there is more protein available to be presented and recognized by the T cells. So at the time of the study, we wanted to give this information out as fast as possible. At the time, it was an important question, which is the reason why Dr. Fauci mentioned our paper. The main message was, yes, a vaccine works. It's going to work. We choose the right protein. And we have not only B cells, but bonus, we're having also T cells. So at the time, the information was very important. But the more the study was progressing, the more on our ends we were asking, okay, so this work when we look very closely after infection. The sample that we analyzed were 1 month after infection. What happened if we have a bigger picture? What happened if we expand this to a larger cohort? That's what we did, and that was a study that went throughout the entire 2020 until the beginning of 2021. We analyzed over 99 subjects, and we fairly tried to assess the protein response of CD4 and CD8 T cells. So here is a recap of our results, and I'm going to try to guide you through it. Each column here represents one of the 99 donors that we have been studying, and each of the rows represent one of the proteins of the virus. So naturally, you can already visualize that the response is focalized in 3 main proteins, the spike, membrane, nucleocapsid, that they keep popping out, but they're not the only ones. Now we start to appreciate that also other non-structural proteins have an importance in the CD4 T cell response, and that applies also in the context of the CD8. Another question that was raising, the more scientific community was trying to really understand the importance of T cells is how broad is the T cell response. So some people may be focusing on specific HLA or specific antigen and so forth, were kind of postulating the fact that the response was narrow, meaning, yes, one donor is going to recognize just some portion of the protein. We don't really know how broad the response is per individual. So we decided to analyze this data in a different way, and we asked how many proteins per donor were recognized. So this is the results that I'm showing you over here, and you can appreciate that on average, the positive donors recognize roughly 3 antigens. And in the context of the CD8, it goes narrow up to 4 antigens. What does that mean? This means that if we're interested in expanding to second generation vaccination, and we are interested in adding additional antigen, that is possible because in reality, once a donor encounters the natural infection, they naturally tend to recognize more than 1 antigen per se. So in conclusion for this part, when we want to look at the overall picture of the immune response against SARS-CoV-2, and we want to have an idea of how the general population respond, when you look at mild non-complicated SARS-CoV-2 infection, therefore the majority of the people that actually get exposed to the virus, we do see a strong adaptive immune response, 100% show antibody response. I haven't shown you this data about the antibody because I guess there are several other groups that show even more in depth than us how the antibody response is characterized during the infection. What we show, and I show you today, is that the majority of the donors have a CD4 response, and 70% of these donors also mount an efficient killer response. So overall, we have a strong response against spike. That was great news for vaccination. And we know that there are other proteins that are recognized and can be used. So up to now, the first question we wanted to understand is how general population behave in the virus. The natural question that came up to us and also other scientists is, okay, but what does make people sick? What is the role of T cells in disease severity? So several fantastic scientists have been focusing their efforts on it, and we were not the only one. I'm just going to show you 2 main papers that summarize the findings of several scientists. And the main concept there is it's a matter of timing, and it's a matter of strong and polyfunctionality. So if you have a response that comprises antibody, killer T cells, CD4 T cells, and they happen fast at the beginning of the infection, you have a mild course of the disease. If you start having a delay in kinetics or lacking one of the components, that's where you have more severe disease. And of course, this was not the only component. As we all know, also age kind of exacerbated the problem. And in fact, the older you are, the higher is the risk that you develop severe disease. So basically, based on these 2 main core papers and also others, if you wanted to recap what's happening during SARS-CoV-2 infection, in a general viral infection, now I'm going to break this. We have the innate response, we have the adaptive, but in reality, we know that the adaptive immunity is composed by antibody and T cell response. They should both increase slowly and be able to go steady and control the virus. So in an average SARS-CoV-2 infection, that's exactly what's happening. You do have the initial viral loads. You have a peak of innate response that needs to go down, otherwise it's too much. The viral load is a little bit delayed, but is not completely removed. To have that, we need to have an efficient response exerted by the adaptive immunity by both T cells and antibodies. When this doesn't happen, the viral load goes crazy. This is what causes severe disease. And the innate immunity is not reduced as it's supposed to be. Even if you have an antibody response, if the T cells kinetics is delayed, they're not able to limit the spreading of the infection. And that is kind of an important point, because in mounting an efficient immune response, you're also making sure that at the second encounter, you're also able to re-recognize this virus and control it better. And in fact, in an acute adaptive immunity, when you get infected, and that applies also during a vaccination, when you get vaccinated, you're going to mount an antibody response as well as helper and killers. And then this first exposure will induce a memory response. So you're gonna still have antibodies and you're gonna have memory B cells that are able to remember this is the type of antibodies you need and also specificity. And that accompanies with the memory CD4 and CD8 T cells that are able to recognize faster the infection and limit the spreading. And this is great, but how long does it last? So this was a question that we were very interested in, and I guess it's a question that is keep pressing and is actually connected to the fact of how long is gonna last the immunity if I get a vaccination. So clearly the virus has not been going on for enough time to make an assessment. We do know that the cousin of SARS-CoV-2 like SARS-CoV-1 has shown memory response 17 years after their exposure, so this is great. What about SARS-CoV-2? So when we performed this study in our lab, we wanted to assess the different component of the immune system, because as it's important to stress, it's not only about B cells, it's not only about T cells, this is a teamwork. That immune response work each piece together to try to fight the virus in different ways. And in fact, what we do see is that the different components are present and also they're polyfunctional. And this polyfunctionality is maintained up to 8 months after exposure. What about T cells? So in the context of the T cells, you should remind yourself that we already started and not all the people actually have a killer response. 70% of the donor has it. So you do observe a slight decline up to 50% for the killer T cells, but that's not the case of CD4 T cells. They remain steady up to 8 months. So this is good news and is a good promise that indeed the immune response, once encounter the virus, build up a memory response that is able to last long. How long? That's hard to say. There's still study keep going and we don't have a precise number right now. So just to conclude this part, we have seen up to now how the immune response behave in the general population. When we look at the immune response against SARS-CoV-2 in relation with disease severity, there are different points that are important to bring up. The neutralizing antibodies are critical, and that is a component that it's important to say. A lot of scientists sometimes tend to just go over antibodies or go over T cells. No, they both are extremely important. Neutralizing antibodies are critical in preventing a cell to be infected. But alone, they're not sufficient to control the disease severity. And that is because, as I was explaining before, if the virus escape antibody, even a little bit, once they infect the cells, they are completely invisible to the T cell response. So this is where the T cell response is required. The T killer to recognize the infected cells and the helper to help in the coordination of the overall immune response. These different components, when they work together, they're able to induce an efficient immune response and a memory response that up to now we know lasts 2 to 8 months. And who knows, maybe it can last more, and more studies are needed to actually address this point. So up to now, I give you an idea of how the T cell response respond in general. But I promised you at the beginning of the talk that I'm gonna go more into in detail at the molecular level on how actually T cell recognize these viral proteins. So the original picture that I show you is gonna complicate a little bit, so let me guide you through it. Assuming that you have an infected cells, all nucleated cells express what we call HLA molecules class 1. These class 1 molecules are the ones that are recognized by the CD8 killer T cells. So virtually any cells that is infected is able to chop a piece of the viral protein presented to the HLA molecules, and the CD8 T cells are able to recognize it and kill the infection. The CD4 T cells, they are coordinating, so they're a little bit more picky, and they don't look at all the different cells, but only the ones that are presenting professionally the antigen. So these are called the antigen presenting cells, so APC, and and only this type of cells present a different type of HLA, class 2, and the class 2 is recognized by the CD4 T cells. Disregarding class 1 or class 2, the portion of the viral protein that is recognized by the T cells is defined as epitope. And the difference between the two is that while the class 1 accommodates very short peptides, the class 2 are able to accommodate larger peptides. So in general, the CD8 T cell response are really more dependent on the different alleles of the HLA, while the CD4 T cells have more flexibility to accommodate larger peptides. So even in the context of variation of the virus, for instance, and so forth, the class II are gonna able to accommodate the response better. So in this context, up to now, we have been investigating the T cell response at the protein level. But in order to really understand what are the portion of the proteins that are recognized, we need to add the complication factor of the HLA alleles. So the HLA is a molecule that is one of the most variable molecule. Each individual express different loci, they are expressed the alleles in a concomitant fashion, meaning that you can have up to 6 to 10 different combinations of HLA molecules in the surface of your cells that are able to present the antigen. So we decide to investigate more in depth what are the epitopes specifically recognized by these T cells. And this is the work that has been done by Alison Tarke, a very promising grad student in our lab. So what she did was, once we identified what were the proteins that were specifically recognized by CD4 and CD8 T cells, she started to test all the different peptides composing these proteins, taking into account the factor of the HLA. And the findings that we had in this case were very striking. So in the context of the CD8 epitopes, we identified 280 epitopes. In the majority of the case, those were recognized by multiple donors. And the most frequent HLA class II alleles were able to bind those. And as I said, this capability is due to the fact that HLA class II molecules accumulate larger peptides, so they also are less sensitive to the different amino acid variation. For the CD8 T cells, the story was very different, and it really depends on the HLA alleles that were studied. And in fact, we were having extreme case scenario like A3201, where the frequency of response and the strength of response was very low. And then we were having cases such as 0101, where you didn't have that many frequent people recognizing it, but when they did, the response was very strong. Or the other way around, alleles like A2601, where every time that a donor that expressed this allele were encountering the specific proteins, the likelihood that the frequency of response was high is high, but in terms of magnitude, the magnitude of the response is very low. So overall, this suggests that the CD8 T cell response is really depending on the HLA. Therefore, it is important to study the different allele, because that takes into account the different geographical background of the population study. And this part, I'm gonna expand it a little bit more in a couple of slides. So up to now, I show you the response in terms of the HLA. What about if we go back and we plot this data, taking into account the protein instead? So we did that, and I'm gonna show you only the data about the spike protein. And the response was quite striking because when we look and plot the epitopes recognized by the CD4 T cell response, it appeared evident that the response was not uniform. There were actually some regions that were highly recognized and some regions less. Indeed, there were clear immunodominant patterns and very defined portion of the protein that the CD4 were able to recognize. And interesting enough, the RBD domain of the spike protein, that usually is the highly most immunogenic part recognized by the antibodies, is poorly recognized by the CD4 T cells. So this means that when one wants to design a vaccine, if you only focus on the portion recognized by the antibodies, you might miss the T cell response. So that's a component that is always important to take into account. Is that true also for the killer cells? Not really, because the variability of the HLA is so wide that the likelihood that at least one portion is going to recognize by one of the allele is higher. So all the data that I showed you up to now is a recap of up to 100 donors in which we try to test the different HLA alleles, but we were not the only one actually trying to identify epitopes. And the scientific community has joined their effort to actually understand how the T cell recognize this virus. And there is a database, perhaps many of you are aware of this already, that is kind of collecting all this information. So the immune epitope database is a database that actually allows you, not only in the context of SARS-CoV-2, actually you have a resource of experimentally derived epitope information coming from different sources: infectious disease, allergen, autoimmune disease, transplantation, cancer, and so forth. So this database has the power to collect data that are published. Those are curated references available in PubMed, also in bioRxiv now. And they take into account information coming from different hosts. So what we decided to do was to expand the studies that we had experimentally by also incorporating the information that had been experimentally defined by other studies. So that was the results of the meta-analysis of up to 25 studies and more than 1,400 epitopes overall. And here I'm gonna show you a little bit recap of this review that was out actually a couple of days ago. And when you start looking at the picture and taking into account all the studies that have been done, they are very similar to what I showed, but here you could even appreciate more that the antigen recognized by the CD4 and CD8 T cells are not only against spike. And in fact, when we focus on the CD4 T cells, while the spike is the major antigen, there is also a good chunk of response that is against nucleocapsid membrane and an sp3 protein. In the context of the CD4, this is even more interesting because it is true that the response against spike is the majority, but this is slightly higher than the response against NST3. So this means that targeting only structural proteins, that does not necessarily means that you are capturing the entire response of the T cell response. In some cases, particularly for the CD8, non-structural proteins can have an important effect in the response of the CD8 T cell. So if we compile all this data again, mapping in the different protein, do we now see a uniform pattern or not? And the response is not. Really, the CD4 and CD8 T cells, they have their preferential sites. So when we look at the CD4 T cells again in the context of spike, we do still see the same pattern that we observed in the context of the study I showed you before, with the majority of the region in the N-terminal domain of the S1 or in the C-terminal domain of the S2, and a poorly recognition in the RBD domain, where the CD8, they recognize all over the place. This is also true, this immunodominant region evidence, not only for the spike protein, but also for other protein. And in fact, you can see that actually the membrane protein is fairly recognized all over, while the nucleocapsid clearly is recognized in the NTD and CTD domains mostly. In the non-structural protein, this is very interesting because we do see a dichotomy between the way CD4 and CD8 T cells recognize the protein. And in fact, CD4 T cells recognize very small defined region of these non-structural proteins, while the CD8 recognize them all over the place. And actually, CD8 T cell response recognize more NSP3 and SP12 than CD4 does. So all this data up to now, I have been discussing about the antigen, but I hope I have convinced you that when you look at T cell, it's not just a matter of antigen, it's also a matter of HLA allele. So what we did was also to understand what are the HLA restrictions for the SARS-CoV-2 epitopes. And unfortunately and unsurprising, we noticed that there was a heavy bias for some specific HLA alleles that are prevalent perhaps in the Caucasian population over other ethnicity. So this kind of show a very clear gap of knowledge. It's important that we start focusing our studies more and more to look into the different general population and different ethnicity, because this is a worldwide virus, this is a global pandemic, so we have to make an effort to actually be comprehensive in our studies. So in conclusion, when we want to zoom into the T cell response against SARS-CoV-2, the main take-home message are that T cells really recognize different protein region, and the fact that they recognize some specific region is depending on the HLA molecules that are addressed and analyzed in the specific context. CD4 and CD8 T cells have a different way to recognize. This is a little bit dictated by the type of HLA molecules that recognizes, and also by the fact that there is a different variability between the different class molecules to molecules. And finally, if you want to design a vaccine and you focus on your target, just looking at what the antibody recognized, you're missing a part very important, the component of the T cell response that are able to induce an efficient viral response. So when you want to design a vaccination and you want to take into account the entire adaptive immune response, it's important that you don't assume targets just looking at one side of the picture. So with this, one could say, okay, good. You look at the T cells, you show that they're important. We got that. You show which protein are important. We understood that. And you also make the fine point of looking at the different immunodominant regions. So we're done. We're good to go. No, because the SARS-CoV-2 actually throw us a curveball and it starts evolving. So the SARS-CoV-2 has been evolving quite a lot in the past 1 year, and more some of the variants start raising concern because they start affecting some specific response or component of the immune response. So clearly, the data I'm going to show you today are a little bit outdated in the sense that, as we all know right now, there is the B1617 that is like strongly replicating in India not only, and is causing a lot of problem. But just hear me out in this talk, because whatever I'm going to show you, we hope is going to be able to be transposed also to other variants in the future. So I'm not going to go over exactly the different variations also because this is evolving very rapidly. What I'm going to just try to let you appreciate is the fact that the antibody response is really changing depending on the variants. It also depends how you're studying the antibody response, because clearly the antibody response can be against the RBD domain, can be monoclonal, can be polyclonal. And depending on the variant that is analyzed, we do see a difference in neutralization that is more or less pronounced depending on the variant that has been particularly addressed. With the B117 that has shown like a minimal reduction in neutralization, to the B1351 and the P1 that are summarized by these three specific mutations, where there is still more consistent reduction in the neutralization. Although the level of neutralization in the majority of the cases are still albeit what is considered a positive response, and that is supported by the fact that several studies of vaccination also in countries with these variants are endemic, do show that there is still efficacy in protection against this variant. But in this study of the variant, one component that was missing at the time, and once again we were wondering, was what about T cells? How are T cells behaving with the variants? So to do that, I'm going to try to give you more of an explanation of the experimental approach, because it's a little bit more complicated than what I show you up to now. So when we talk about mutation, particularly when we're focusing about antibodies, everybody is looking at the spike protein only. But in reality, these variants are composed by mutations that occur throughout the entire proteome of the virus. So what we did to try to assess the comprehensive capability to react against the variant was to compile the list of variant mutations concerning the different variants at the time of our study. And we tried to generate peptide pools for each protein. Why? Because it is true that you have a mutation in a specific point. It is also true that your T cell response is against the entire protein. So if I only focus my study in understanding the mutation, I am losing the big picture. Do I still have a T cell response that is able to recognize this variant or not? So this is why what we did was to take the entire protein, spike and not only, and what we did was just we swapped peptides that were mutated with the actual mutated peptide, and each of these pools then were corresponding to the different variants in study. So what we want to understand, we were trying to put ourselves in the situation of someone that got exposed to the virus before the variant occurred, or someone that is receiving the vaccination. So what we did was we tried to collect samples before October 2020, in California, that we know at the time, the prevalence of the other variants was very mild. And we also decided to study donors that were vaccinated. And at the time of the study, the only vaccine available were the Moderna and Pfizer. So we analyzed donors after the second dose of vaccination. And I'm reiterating this point because it's going to be important the more we go through the study. The spike protein is clearly quite mutated, but it's not the only one. We do have mutation throughout the entire genome. And that is an important point. For instance, in the context of the B117, we have a codon subset of 8 disappear, it's not there anymore. So we completely lost the response against an entire protein. So what are the results that we have? So I'm going to show you just the results that we have available on the bioarchive. In the meantime, we have been expanding this data considerably. We double up all the cores, and we do hope that the data are going to be accepted soon. So we're going to be able to show the entire data set to the wild community. So here, if you look at the response against the ancestral, this is the response of the donor after getting exposed to SARS-CoV-2. When we try to look at the response of the same donor stimulated with the different variant peptides, we do see that overall the response is very similar. You might have some specific case of a decrease of a single donor, but in the general population, the response is unchanged. And that is true for the CD4 and the CD8 T-cell response when we look at the spike protein. It is also true when we start to compile the total magnitude of response. So with the total magnitude means that we look at the single response for each of the protein, wild type and mutated, we sum them together and we evaluate the overall response. So even more, if you get the infection before, so you have seen the different protein of the virus, your T cells are gonna be able to cross-recognize the different variants. And that is true again for both CD4 and CD8. So this is a great news. This means that if you have been previously exposed, you are able to cross-recognize the different variants. And that is the reason why, even though these variants are keep occurring, we do have the capability of the T cells to limit the spread of the disease. So you might have still the infection, but the disease severity of this infection is not as strong as it could have been if you've never been exposed to the virus before. So this is the good news for the people that unfortunately got exposed to the virus. Is that true also for the vaccines? It is. And in fact, when you receive a vaccination with the spike protein, you are able to cross-recognize the spike protein of the different variants. And this is true for both CD4 and CD8. So this is at the overall protein level, and this was an agnostic type of strategy. So I am not factorizing the fact that there is the HLA component and just taking into account the entire protein. What happens if we start taking into account the actual epitopes? So we went back at the time to the epitopes that we identified in our previous study, and we tried to understand how many of these epitopes were actually affected by mutation. And nicely enough, 92% of the total epitopes recognized for CD4 and 98% for the CD8 were conserved, so they were not affected by mutation. And that was for all the proteins. What about the spike if you only want to take into account vaccines? The picture doesn't change. You still have 90% and 96% of the epitope previously identified still conserved, meaning that the variants are still mutating, but those mutations are not sufficient to completely disrupt the overall T-cell reactivity. And then among the ones that were mutated, we even made a step forward and we were trying to understand how these mutations were affecting CD8 T-cell response. And the majority of those were at either neutral or even increasing the binding capability, therefore suggesting that most likely even the mutated peptides can be cross-recognized by the immune system. So to conclude this part, the antibody response, as I was mentioning, show a decrease with the variants, but they're still present, and that is an important component. The T-cell response instead are largely unaffected in the variants we analyze, And this is true whether we look at previous exposed donors or vaccinees. So one can speculate that the vaccine can induce a sufficient significant T-cell response that can be able to cross-recognize the variants and actually still protect from severe disease, which is what we observe actually when we look at vaccination. It is true that during with the variants, we do see a reduction in neutralization. It is true that some people can experience reinfection, but 100% of the donors are still protected from severe disease and death. And so that can also be due to the contribution of the T-cell response. So there is, of course, a big question that remains unresolved. And what about the other box? What's happening with the B167.617 variant? And of course, despite the fact that everybody is trying to work on this, there is a little bit of delay because we need time to produce these peptides and actually address this question. I'm gonna just leave you with a very small thing that derived from our study. I just went through it very fast, but in reality, one point that we observed is that there was like some pre-exposure activity against SARS-CoV-2 in donors that have never seen the virus. So we have investigated this a little bit more, and we have shown that actually this is related to previous exposure with other common cold coronaviruses. So that suggests also that in the vision of designing a vaccination, one can envision a pan-coronavirus vaccination that take into account portions that are shared between previous common cold coronavirus and SARS-CoV-2. And finally, what do we do if a new outbreak comes? So we have now tools that are able to predict T-cell target at the beginning of the pandemic. And that's something that we started at the real beginning of 2020. That's what allowed us to actually start straight away and analyze T-cell response in SARS-CoV-2 subject. And that is due to the progress that the bioinformatic analysis have been doing throughout this time and the presence of IDB. So we were able to predict T-cell targets based on the information we knew about previous epidemics of SARS and MERS, and that allowed us to actually focus our results on the candidates that were the most important. And later studies have actually shown that the predicted candidates that we predict at the beginning were indeed the immunogenic in SARS-CoV-2. So this is promising and shows us that we indeed have some tools to actually start studying a new epidemics if it occurs. And with that one, I have shown you the study of many, many people, but science, like the immune system, is a teamwork. So everything that I show you is actually the heavy work of many people, many PIs related at the La Jolla Institute. I would like to thank Daniela Westcott, Alessandro Settes, Shane Crotti, and Bjorn Peters, the 4 main PIs that have been driven many of these studies. And the majority of the studies that I show you have been actually done by Alison Parker, our grad student. And with that one, I hope you enjoyed the talk, and I'm happy to address any question you might have.
Alba, thank you so much for such a great presentation. And first of all, thank you for accepting our invitation so graciously. And we were really excited to hear about the role of our unsung heroes, T cells, from someone who has dedicated her entire career studying T cells. So thank you so much for that. And also, I would like to add that the importance of T cells has been highlighted by the observation that some people with A-gamma globulinemia, where they cannot make antibodies, or people who are on rituximab or B-cell depleting agents we use in CLL, chronic lymphocytic lymphoma, or rheumatoid arthritis, have had relatively mild cases. So that tells you the importance of T cells right there. So thank you so much for adding to our understanding of T cells. And if anyone has any questions, I'll just look at the chat box. So there's a question. So knowing the epitopes that are recognized by T cells, would you have predicted the suboptimal immune response, especially by vaccines, given the mutations along S gene?
So that's an interesting question. So first of all, I have shown you that the majority of the spike epitopes are actually not affected by the mutation, therefore suggesting that spike is still a good candidate, particularly for the T cell response. In terms of predicting yeah, so I guess this question is very tricky because like it's assuming already that there is a suboptimal immune response, which I'm not sure that is the case. I think what we can say for sure is that if you know the protein sequence of a virus and you want to know which portion are going to most likely be recognized by the T cells, we do now have some prediction algorithm that can help us guiding through the process. That, of course, is important to be supplemented by experimental work. In terms of vaccination and mutation along the S gene, I guess there is an additional component to take into account, which are mutation due to the escape to and the antibody response, which kind of can give the suboptimal immune response. So clearly, to overcome this issue, one might consider to add to the spike protein also additional antigen targets that are specific only for T cells, so you can ameliorate the problem from a T cell point of view. For someone who has reaction to previous vaccination, has had COVID-19 before, can a substitute vaccine be taken? Can I have this question explained a little bit better? If I understood it correct, the question is, if you had a previous reaction to a vaccination or had COVID-19 before, can a substitute vaccine be taken? What do you mean with substitute vaccine? There are different vaccines circulating worldwide, and depending on the country, there are different licenses. Studies shown that it's perfectly safe if you have COVID before that you can have a COVID vaccination, even both doses, and that's still what the guidelines suggesting. So I don't think I follow the question.
All right, so with the interest of time, I would like to.
Oh, that's
All
a
right,
very
so
nice
there's another
one.
question.
Does T cell activity to SARS-CoV-2 in non-exposed individual would have protective immunity? We do not know that yet. We hypothesize that actually having pre-exposure to common cold corona and having some memory T cells already ready to go would have actually helped you in protecting. However, when we look in depth in the epitope repertoire that was given by this cross-reactivity and the one given by the SARS-CoV-2 infection, we do see that there is a little bit of overlap, but once you get exposed to the virus, there is a whole new world that is happening. So our hypothesis is that if you get exposed to the virus, you might have a kickstart and maybe milden up the disease, but I wouldn't say that this overall protects you. And it really depends on the T cell response that you have. I would like to point out that 50% of the donors that we analyzed were actually not having response against common cold corona. So the reaction was factual palsy post-vaccination. Okay, so the question has been explained a little bit better. I apologize, I don't have the clinical background to actually reply to a side effect with the vaccination. Why a person gets repeated infection? What could be a problem in developing immune system in such a case? Excellent question. So what we have been discussing with you before is the fact that when you have disease severity, what happens a lot of time is that you have an uncoordinated immune response. So for instance, you lack of one of the components, you can lack antibody response, you can lack one of the components of the T cell response, you can have a delayed in kinetics of the T cell response. So these, based on what we have seen, can be the major reason why you are not controlling the virus. So that also means that you don't induce an efficient memory response, and that could also lead to repeated infection. The plasma therapy, as I said, this is more a clinical related question. So I am pretty sure there are people that have more experience than me in the field and can reply better to this question.
Okay. All right. So with the interest of time, we just have a few more minutes now. So I just want to ask a few questions for you. So regarding the epitope, so you guys predicted all the epitopes, so you went over that. So can you describe to our audience what a megapool is? Because you use the megapool approach to study the T cells. in briefly, can you just explain what a megapool is?
Yes, of course. I have one slide perhaps that can help explain the concept. So what we call about megapool is taking into account the assumption that when we consider the protein of the entire pathogen, all the possible peptides, and the combination of these with a different HLA, we perform a filter done by prediction or experimental defined epitopes to actually select a smaller list of peptides. We call it megapool because we perform what we call a sequential lyophilization to be able to go from this entire complexity to a very small reagent, and that has the advantage to have a synergistic effect when you look at the different epitopes, a good HLA coverage and protein coverage, a minimized DMSO toxicity, and the advantage to be used with small biological samples. So our lab has devoted their effort to have these freely available to the entire community. We have been sharing this to up to 110 collaborators worldwide, and that actually allowed to kind of like use a similar approach to dissect T-cell response in different geographical locations. And more studies with this collaboration are coming out to actually show if there are differences between different geographical backgrounds.
All right, thank you so much for explaining that. And one final question I want to add is, so just regarding the vaccine, so you touched upon that as well, and it's regarding the next generation of vaccines, because almost everything right now is based upon spike. I think there's only one vaccine or one or two vaccines in progress in the making right now that includes M protein also, which is still in like phase 2 trials, I believe. So what do you think the next generation of vaccines should incorporate? Do you think it's M protein? Because from the papers, we can see that M protein also, M as in membrane protein, also elicits a lot of T-cell response. So that's for immunodominant too. Would that be a right antigen to add in the next generation of mRNA vaccines or something?
So I guess it really depends where we want to go with the second vaccine generation candidates. So for instance, first of all, we want to take into account also the effect of the proteins at the antibody level, and the data on the nucleocapsid protein are there are different opinions about the antibody response against these proteins. So that's something that should be taken into account also with the antibody counterpart. The membrane protein is clearly quite strong, gives quite strong response. It is also true that it's poorly cross-recognized with the different other component or other coronaviruses. So if the goal is to design a vaccine that is more of a pan-corona vaccine and is able to induce a response across different viruses, then the choice of the candidates will also have to take that into account. If instead we want to just do a vaccination that is against SARS-CoV-2, then indeed the spike is a good candidate, nucleocapsid is a good candidate, the membrane protein is a good candidate, and we have seen also some non-structural proteins are also good candidates that can be evaluated.
Okay, and if we were to incorporate like M gene in there, would it be better to incorporate like in short peptides or just the entire protein to induce a better T-cell response?
So that's a good point. If we want to just narrow down to some immunodominant region, what is important to do is to take into account the HLA variability. So one should have to make a feeding study to see actually what are the portion of the protein recognized worldwide, taking into account the different genetic backgrounds. So an alternative approach just to make sure to incorporate all these immunodominant region is to use the entire protein, but it really depends on the vaccine candidates and platform and the capability they have to accommodate larger or smaller proteins, I would say. Sure.
All right then. Thank you so much, Alba, for everything. So we had a great time here. We learned a lot. I'm sure everyone learned a lot and enjoyed this presentation. Yeah, and thank you so much for your time. Ciao and grazie.
Prego, grazie. Thank you very much. It was a pleasure being here.
I would love to have you again in the future if your schedule permits at some time.
Of course, yes. Thank you very much.